we are a consortium with 5 research teams, plus a company helping us with the management and the outreach. We are interested in studying complex chemical reaction networks, and for that reason we have involved in the team groups with expertise in chemical synthesis and metabolomics analysis applied to CRNs, also cheminformatic tools, and very important, I forgot to say, so the aim is basically to implement in a physical platform, which is basically the CRNs in a microfluidic chip, reservoir computing capacities. And Professor Wilhelm Huck from the Radboud University in Nijmegen will explain in the first talk much better than myself about this idea. So microfluidics, having highly controllable and reproducible microfluidic chips is critical to achieve this goal, to study the CRNs, and this is the purpose of today, bringing together Emmanuel Loerster from IBM, who's an expert on this aspect, but also some external experts, just to engage and to share ideas and discuss about this. Okay, so the final goal or the long-term vision is, of course, to construct brain mimicking computing devices that utilize complex chemical reaction networks as information processing systems. This is the program. Professor Wilhelm Huck from the Radboud University in Nijmegen will start, followed by Emmanuel Loerster, sorry for the pronunciation, from IBM, Zurich. And our external speaker, I really appreciate that Professor Klaas Hjort could join us today from Uppsala University in Sweden. So the best is to start with the science. I will give now the screen to Professor Wilhelm Huck, and thank you very much for your assistance to this webinar. I will stop the sharing.
Okay, thank you, Andres. I will take over from here. I hope that everything is well visible. And then I will try in the next 15 minutes or so explain to you what we're doing with reservoir computing using chemical reaction networks. And so on my screen already are the main contributors. It's Mathieu Baltussen, Thijs de Jong, Will Robinson, and Quentin Duess. So our group is interested in chemical reaction networks. And for those of you who are not familiar with them, think of a living cell. There are many, many chemical reaction networks in there, ranging from metabolic networks to signaling networks. And these are typically consist of enzymes. And so you can try and build such kind of networks in vitro, in a flow reactor, by designing such a way that your enzymes are connected via the enzymatic reactions that take place. But today I will be talking about small molecule reaction networks that in a way emerge by just reacting simple compounds together that then form a huge network of chemical reactions producing lots of different compounds. And the reaction that we will be studying today is the so-called Formose reaction. So it looks very, very complex, but actually if you would study this in some detail, then you start actually with formaldehyde and a sugar, 2-carbon sugar or a 3-carbon sugar. Here you see dihydroxy acetone. And if you just mix them together under the right conditions, and right conditions simply means you have some calcium catalyst in there and a high pH, so typically pH 14, then within a few minutes you form tens to maybe even hundreds of compounds, where the complexity in terms of compounds is very, very diverse, but the complexity in terms of chemical reactions is actually fairly simple. So the chemical reactions that are taking place is proton transfer, aldol addition reactions, retroaldol reactions that essentially break things up again, and also some so-called Cannizzaro reaction, which is a side reaction in this. So the bottom line that you have to take away from this particular slide is that by doing some very simple chemistry, we generate a very complex chemical reaction network. And we have studied this in detail. This network is very responsive to any change you make in the environment. So if you change a little bit the pH or the concentration of formaldehyde or the concentration of your sugar, then everything changes in this network. So that's very important to remember. Now, the way we study this in this particular study that I'm talking about today is by actually running this reaction in a so-called flow reactor, in a continuously stirred tank reactor. And the speakers after me will discuss this in great detail. For now, you can say, well, it's a very small reaction flask. You have the ingredients going in, formaldehyde, calcium chloride, sodium hydroxide, and a sugar. And then what comes out in this particular instance is run directly into a mass spectrometer. So we're not actually characterizing the precise nature of what comes out in this instance. And so what that allows us is to measure continuously for a long period of time, say 7.5 hours. Every half a second, we get a data point, and the data points look like this. You see this on the right part of the screen, where we follow about 100 different traces, where each trace here is a unique ion that comes out of the reactor. So this can be different compounds, but it can also be different calcium complexes, for example. And so these 100 different traces over time, that is going to be our input data into the chemical reservoir computer that I'm going to explain next. So this is the experimental setup. Now, what are we actually going to do is the following. We're going to say, well, when we have this chemical reaction network, that is a so-called chemical reservoir. We're not going to be bothered too much what is happening there. We're going to say, well, there's going to be different inputs, formaldehyde, dihydroxyacetone, maybe temperature, maybe pH. Those are your inputs. And what comes out at the other end, we're going to use to do so-called classification problems. So I should be able to classify my environment. Or if you think about a more simple problem, I want to distinguish pictures of cats and dogs. That is one thing, that's a static system. The other thing that we will be studying is dynamics. So I can constantly change my input and constantly track my output, and then map that constantly changing input onto the system dynamics and understand sort of constantly changing environments. So those are the kind of tasks that we will be studying. Now, how will we do this? Well, it's going to be a little bit complex, so this is an important slide. So what we will consider is a so-called reservoir computer. So typically a reservoir is constructed in silico, where people make some piece of software where lots of different so-called agents are all connected to one another in some kind of random nonlinear fashion. That reservoir, in our case, is the foremost reaction. Now, into this reservoir, you have a so-called input layer. So for us, this can be concentrations of formaldehyde, the sugars, temperature, etc. And as you can imagine, they will change something in the reaction network. So at the other end, something will change. And so the readout layer in our case are these 100 traces in the mass spectrometer that I have shown. So far, so good. We have a certain input, we have a reservoir, and we have a certain output. And for the reservoir computing, it doesn't really matter what happens here. This is a black box. As long as we know it generates a complex state space, it does, because we have 100 traces coming from just a few inputs. It's highly nonlinear because these chemical reactions are all over the place. There are feedback loops in there. There's a fading memory because if I flush my reactor, obviously I continuously refresh everything. And in the end, I can measure each of these signals separately. So this is our sort of setup for a so-called physical reservoir computer. Now, the next thing we need to do is, of course, to say, well, in the end, this list of peaks that I get out of a mass spectrometer, that must mean something. So how do we do that? Well, you get a list of peaks. That's your chemical output. So this is compound 1, compound 2, compound 3. And for each of these compounds, you have a certain peak height. Now, if I want to do a classification, then at some point I have to say, well, that kind of particular output that I had for this particular reaction should either resemble a 1 or a 0 or a certain value. Well, it obviously doesn't, because if I sum up all these values here, it doesn't add up to 1 and certainly not up to 0. So I need to find a set of weights, a filter if you wish, that maps onto every one of these outputs in such a way that the combined output, if I multiply the weights times the height of the peak, starts to get closer and closer to the value that I would like to have. And you say, that sounds very complicated, but actually it's not such a complicated problem. It's a linear regression problem. Your computer can do this very, very rapidly. So very rapidly, I find this filter that will tell me that applying this filter on this data will give me the value that I want. Okay, so that is how it works. This is how you train the reservoir. And then you can say, okay, now let's do a real experiment. We change the formaldehyde concentration, we change the sodium hydroxide concentration, and we do 96 or I think 100 different combinations. So for each of these combinations, I have this output where I have many, many different compounds and a certain intensity. And they will all be different, of course. So what I now need to do is I say, well, if this would be my target, and all of these should be 0, and all of these red ones should be a 1, then I need to find one filter for all of the 100 data points that classifies all of these as a 1 and all of these as a 0. And of course, I will not find a perfect filter, right? But I need to find something that gets as close as possible by training it on a certain set of data and then testing it for the rest. Now, this sounds very difficult. It turns out that my reservoir generates data that makes this extremely possible. So on very simple problems where you have an AND gate, so-called AND gate, where you say every one of these should be a 1, every one of these should be a 0. I can do an OR gate or a linear function, but then I can start with more complex, a triangle or an XOR gate, which is a nonlinear problem, or I can make very complex patterns, or indeed I could give it a picture of a cat and a dog and say, well, classify this correctly. And as you can see, my foremost reservoir computer gets a so-called phi score that just tells you how close it is to predicting this accurately. And this is always the first bar in every one of these tasks. And you see that it competes or even outcompetes all of these other algorithms. And these are machine learning, so AI type algorithms that allow you to treat this data as well, but then purely in silico. So in silico algorithms are not better, or marginally better sometimes, than my chemical reaction that is just this mass of chemical molecules. So this is the first demonstration that this principle actually works. And then you say, well, this is nice, but this was just static data, right? I just feed it with a certain combination, as I showed you on the previous slide, a certain combination of formaldehyde and sodium hydroxide. I wait till the reservoir stabilizes and then I measure this compound composition. I can also say, well, I could constantly change things and then measure things that are constantly changing in time. So we have done this. We constantly change the input into the reservoir and then we train it. And what do we train it on? Well, we could say in silico, I can download the rate equations for the carbon metabolism of E. coli. This is known, you can find it on the web. This has 92 different chemical reactions, 87 different substrates. So you can imagine this is a large set of ODEs, and if you feed glucose into this model, you essentially get 87 different intermediates into your citric acid cycle, etc., etc. And so you can calculate, if you change the glucose concentration over time, how each of these 87 different substrates changes over time. And so I can then say, well, if I do this in silico, I can sort of simulate all of these 87 different substrates, but I could also do a similar input variation into my reservoir. So that's not glucose, but just formaldehyde concentration, fluctuating in, training it, and then see if I can match my foremost reservoir to my in silico calculations. If you do that, then this is the actual network that you download from the web and you can simulate. So you have a certain input. In this case, it goes into your network over here, and then everything propagates, obviously, over time. And then what you can do is you can train so the red line that you see here is how each of the intermediates, pyruvate, 3PG, 8MP, fluctuates over time if I have a certain input into this very complex network. That's the red line. I then train my reservoir and I get a minimum error after a while. So I find a set of weights that predicts accurately how pyruvate, 3PG, and AMP should move over time. That's the yellow line. If I then stop and I'll just let my system continue, then as you can see, for the next hour or so, my chemical reservoir can predict the changes in concentrations over time, given that this would be my input concentration into this network. So my reservoir doesn't know any ODEs, but it has learned how apparently this system works by mapping it onto the predicted values for the first 30 minutes. And you say, well, that works for 3 compounds. There were 87 compounds in your mixture. How is that going? Well, this works for all the 87 different components in my network, whether they are very, very low concentrations, low millimolars, or high millimolars, sort of high micromolars to high millimolars. All of these concentrations map very, very accurately using my foremost reservoir. So that is what I was trying to tell you, was going to try and tell you this morning. So to wrap up, this is the first ever example of a chemical reservoir computer. This was made possible because the foremost reaction has this, well, we believe fairly unique structure where there is all these feedback loops and nonlinear interactions where everything is connected to everything. Because very few chemical reactions produce all these different compounds, and any change in the network in terms of input leads to the network reorganizing completely. Now, this is not only the first example of a chemical reservoir computer, it is also, I think, the most powerful physical reservoir computer reported so far. So there's nothing out there that shows that physical reservoirs can predict these kind of complex dynamic systems with such high accuracy. Now, that is very nice, and one of the reasons why I'm presenting to you here today is that now, of course, we need to think about how to go further, because I don't think there will be a foremost reservoir computer replacing your laptop or your phone or something, right? Because it is an actual physical system where chemicals go in and out. So we have to think about what should this computer actually calculate, and how do we map this to some kind of chemical function, right? Because if you go back to living systems, they might also have this kind of computer capabilities, but they don't sort of solve sets of ODEs. They just want to know whether it's warm or cold outside, or whether it's light or dark, or sort of wet or dry, if I look outside here in the Netherlands. So your system processes other kind of information and does something else with that information. And that is something that we need to think about next. With that, my 15 minutes are gone, I think. I need to thank the people who did the work. I already mentioned them in the beginning. I would like to thank CORENET for funding of this project, and thank you for your kind attention.